So just a few slides to wrap up and look at some of the specifics for cancer detection in the K-Ras gene. So circulating nucleic acid or DNA molecules. Turns out that when you do have a cancer tumor, you do release some of the DNA molecules from that cancer into your bloodstream. All right. This is just describing some of the mechanisms for that. Turns out overall that when you have cancer, you actually have a higher level of oligonucleotides in your blood, and you also have some, more in our case, that are, or more for our concern, that are very specific to the cancer. One of the things out there is that there are a lot of other techniques for detecting these oligonucleotides. The problem with many of them is that they're not very specific, okay? So if I want to detect mutant DNA, which that's the portion that's indicative of the cancer in someone's blood, all right, if I do direct sequencing, I can only detect if I have at least 50% mutant DNA in that. You go through electrophoresis, HPLC, etc. And in all of these cases, you can get down to actually some relatively good values, but you're talking about a few percent of that mutant DNA that you need relative to the native DNA. If I'm at early stages of cancer, that's likely not going to be good enough, and I have to figure out some ways of doing that. That's one of the things that specific PCR is being used for, is to actually amplify the mutant DNA relative to the native DNA to get it up into these ranges so that you can detect it. From our purposes, we're using surfaces, that kind of probe on the surface that I talked about, and we're trying to up. All right, so if we have the mutant type capture probe on the surface, what it actually turns out is that you only have a single base that changes between the mutant type and the target. So this C is changed to a G when you develop, or when the cancer develops in someone. And so this is the mutant type. It'll hybridize very specifically to this surface. The problem is that it'll also tend to hybridize to some extent to this. All right? The binding constants for these two binding reactions are not all that different. And so we've had to develop a variety of techniques that'll come in and do what's called primer extension. The idea behind primer extension is that what we'll do is take this capture probe, the DNA molecule that we've bound to the surface, and we'll actually selectively extend it. And by doing that, we can actually amplify this difference in binding. You can also put in enzymes that relatively quickly degrade the bases that you're using. If you do that, then again, because this extension is slower, you actually don't tend to do as much of this, and you kind of multiply these factors, and when we extend this, we actually couple the biotins to this directly. So we extend this very specifically, try to develop biochemical techniques that have allowed us to extend it specifically, and then also, beyond that, actually couple the biotins into that. And so what that's allowed us to do is now actually to detect specific mutations on the surfaces. We don't have the selectivity studies yet. I'll show some things that allude to that here on the next slide. This is a pre-amplified surface. We actually use an initiator at this point that fluoresces. So with a nice scanner, we can actually tell where our initiator is before we do the amplification. This is our positive control, basically biotin printed directly onto this. This is the DNA molecule with the cancer mutation indicative on it. And then we go and we amplify that, and you can see the spots in both of these cases. Again, just backscattered light, excuse me, in this instance, it's actually side scattered light, just a digital camera image of this. All right. So one of the exciting things about this then is that we are able to get relative specificity, and yeah, that's what I thought. You can barely see this on these. On the actual picture, you can see it much better. There are two spots here that show up. If you don't believe me, we actually measure the thickness of these so you can actually see them. So 4 spots on this, and then it's exposed to 1 micromolar DNA of the wild type DNA, no mutant hybridization, or no mutant DNA is added to this. And then we have the 2 capture probes, the wild type capture and the mutant capture. And you see that we get almost no cross hybridization and subsequent polymerization. And that's indicated by measuring the thickness of the polymer film that's there. And in this case, we get less than 10 nanometers for both the non-specific capture and the mutant capture. So the techniques that we've developed biochemically are very specific for coupling our initiators to this species, to the surface, and then subsequently getting polymerization. And you can see our wild type capture doesn't give us as much polymerization as the biotin control because we don't have as much of the wild type on the surface as we do the control. But we get significant polymerization in both of these instances, enough that under just camera images and by eye, you can actually see the 2 spots present in those cases. So with that, I'll end, and I'll say that we're able to use photopolymerization to detect these molecules, to do all sorts of different types of biodetection. I focused today on cancer detection, but we're actually interested in using it for a variety of other aspects. Turns out that by combining these systems in our best case scenarios, we can get amplifications for every biotin that's on that surface. We could get 10 to the 11th monomers to polymerize. All right, a huge number, of order of magnitude increase in that. And that allows us to detect about 1 molecule for every 20 square microns. It's a lot of work still to be done, but it's a start, and I think it's a relatively nice start. And I owe that to the students and postdocs that I've had on the project, as well as the funding. Tech Transfer, the National Institutes of Health, and others have supported this research very well and continue to do so. So thank you very much, and I'll be happy to answer any questions. Yeah.
What did you use the 1360 grant for? Which part of this?
So we have in particular used that for what I've talked about here with the last couple slides, where we focused on how do you develop techniques for sensitively and specifically detecting single base pair mismatches, as you frequently want to do in cancer detection.
Okay, well, we have another question. Yeah, please.
So you showed your controls and it looks great. Could you comment on your thoughts on the false positive rate?
So being one that's been affected by false positives on medical tests, it's a difficult thing. And so there are 2 different types of false positives. One we can do something about, one we can't. So we can't do anything about the lack of biological specificity. So our technique, if you gave us the prostate-specific antigen and asked us to detect it in very low quantities, we'd have the same problems with that being nonspecific and giving false positives, because that antigen is not specific to prostate cancer. All right, it's indicative of a lot of other things. We can do absolutely nothing about that type of false positive. All we can do is say, you have this level of this compound in your blood or in your urine or whatever the test happens to be. All right, so that we can do nothing about, and it's something that we need to rely on the biologists, biochemists, et cetera, medical practitioners to give us better molecules that are indicative of the disease state. In terms of the other types of false positives, we're going to have an issue because we're so sensitive. And this is one of the things. So one of the things that I didn't talk about is going into blood and serum and things like that. And we've actually had some very, very promising early results in those complex biological fluids. But that's going to be something that we're not likely to be able to maintain the amplification rates that we have and also not give rise to false positives. And so somewhere in there for a given test, somebody's going to have to decide, as somebody who's going to sell these ultimately, what's the right level? Do I need to detect 10 to the 6th molecules instead of the 10 to the 3rd? And by so doing, can I eliminate a lot of false positives? And absolutely, we can give them all sorts of techniques that then would eliminate everything under about 10 to the 6th molecules. But if you want the really high sensitivity, ultimately you're going to have higher false positive rates because of non-specificity. And we can do some things about that. I mean, the single base pair mismatch is a good example of what we can do with some of those things, but we can't solve the problem altogether. And that's, with every high amplification technique, that's an issue.